Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today, we're going to be covering Chapter 5 in our MCAT Biochemistry series, and this chapter is titled Lipid Structure and Function. In this chapter, we're going to be covering the following objectives. First, we're going to start with structural lipids. This includes phospholipids, glycerophospholipids, sphingolipids, and waxes. These are lipids that primarily serve as components of membranes or protective barriers in cells. Second, we're going to talk about signaling lipids. These are lipids that act as messengers in the body. And we're going to focus on terpenes and terpenoids, steroid hormones, prostaglandins, and the fat-soluble vitamins A, D, E, and K. Third, we're going to look at energy storage. This includes triacylglycerols and free fatty acids. And we're also going to explore saponification. This is the chemical breakdown of fats into soap. With that, let's get started with our first objective, structural lipids. Lipids as a class are defined by their solubility properties. They are insoluble in water, but they're soluble in nonpolar organic solvents, which makes them ideal components for biological membranes. In fact, the most important structural use of lipids in the body is in the phospholipid bilayer. This forms the foundation of all cell membranes. This bilayer acts as a barrier that separates the internal contents of the cell from the external environment. Now, each lipid in the membrane is amphipathic. That means that the molecule has both a hydrophilic region, which interacts well with water, and a hydrophobic region, which avoids water. And this dual nature is what allows lipid molecules to arrange themselves into a bilayer, with the hydrophobic tails tucked inside and the hydrophilic heads facing outward towards the aqueous surroundings. Now, more broadly, one thing all structural lipids have in common is that they contain long hydrocarbon tails made up of fatty acids. These tails, they can vary in length and they can vary in their degree of saturation, meaning how many double bonds are present in the chain. Let's break that down further. A saturated fatty acid has only single bonds between its carbon atoms. Because the chains are straight here and they can pack tightly, these lipids tend to be solid at room temperature. So think of butter or animal fat. That's a good example of a saturated fatty acid. An unsaturated fatty acid, on the other hand, it contains one or more double bonds. And these double bonds create kinks in the chain, which prevents tight packing. So that makes them more fluid or liquid at room temperature. So think of olive oil. So this combination of tail length and saturation really affects how fluid or rigid a membrane is going to be. And that, of course, has really big implications for membrane function. Now, let's briefly introduce the two important categories of structural lipids that we're going to focus on in this chapter. We're going to discuss both in detail in this first objective, and at the end, we're going to have a flow chart that helps us think about things more clearly. First, we have glycolipids. These contain a sugar group as the polar head, and they're joined to a fatty acid tail by glycosidic linkage. Now, the sugar head group, it gives glycolipids a unique role in things like cell recognition and signaling, especially on the outer surface of membranes. We can further classify glycolipids based on how many and what types of sugar they contain. We're going to get to that shortly. But second, we have phospholipids. These contain a phosphate and an alcohol in the polar head, and they're joined to a fatty acid tail through phosphodiester linkages. Phospholipids are the main component of the membrane bilayer, like we said earlier, and they can be further classified based on their backbone structure. With that, let's start with elaborating on phospholipids further. Again, to reiterate, all phospholipids have the same basic structure. They contain a polar head group, which includes a phosphate and an alcohol, and they're attached to hydrophobic fatty acid tails. What defines the type of phospholipid is the backbone that connects everything together. The first type of phospholipid we are going to discuss is glycerophospholipids, also known as phosphoglycerides, and they have a glycerol backbone. That glycerol is linked to two fatty acids by ester bonds and to a highly polar head group through a phosphodiester linkage. Now you can see this clearly in the figure on this page. The glycerol sits in the middle. The fatty acid tails, they extend downward. One is straight and one is a little bit kinked to represent saturation and unsaturation. And the phosphate head group, it projects outward to interact with water. This structure makes glycerophospholipids amphipathic, and it allows them to form the lipid bilayer of cell membranes. Now, the identity of the head group, it can vary, and that determines the surface properties of the molecule. If the head group is neutral, positive, or negative, that's going to change how the lipid interacts with other molecules in the membrane, including things like proteins, receptors, and signaling molecules. Now, glycerol isn't the only type of backbone we see. Some phospholipids use a different kind of backbone called sphingosine, and these are called sphingophospholipids. Just like glycerophospholipids, they have a phosphate-containing head group, and they serve structural roles in membranes. So both glycerophospholipids and sphingophospholipids fall under the broad category of phospholipids. They just differ in their backbone. Now, this leads us to sphingolipids more generally. Sphingolipids are built on a sphingosine or sphingosine-like backbone, and not all of them are phospholipids. In fact, many sphingolipids are glycolipids. That leads us to our second category of structural lipids. And glycolipids, like we said earlier, they have a sugar-based head group instead of a phosphate group. So from here, what we're going to do is we're going to explore the major classes of sphingolipids that we need to know for the MCAT, and we're going to identify them as either phospholipids or glycolipids. First, we have ceramide. This is the simplest sphingolipid. It's essentially the core structure from which other sphingolipids are built. Ceramide has a hydrogen atom as its head group, and you can think of it as the foundation for more complex derivatives. So in other words, it is neither a phospholipid nor a glycolipid. Instead, it serves as the precursor to both sphingospholipids and glycosphingolipids. But on its own, it doesn't contain a sugar or a phosphate head group. Next is sphingomyelin. This is a type of phospholipid. It contains a phosphocholine or a phosphoethanolamine head group, meaning it includes a phosphate group, and that's what classifies it as a sphingophospholipid. Sphingomyelin also contains a phosphodiester bond, and functionally, it's a major component of the myelin sheath, which insulates nerve cells, and it speeds up signal transmission. Then we can move into the glycolipid family of sphingolipids. These are called glycosphingolipids. In glycosphingolipids, the head group is made of sugar molecules, and these are typically found on the outer surface of the plasma membrane. And there they play key roles in cell-to-cell recognition and signaling. Here there are two main types I want to discuss. We have cerebrosites, which contain a single sugar residue like glucose or galactose. And we also have globocytes, which contain 2 or more sugars. Both of these are neutral at physiological pH, meaning they don't carry a net charge. Finally, we have gangliocytes, which are the most complex glycosphingolipids. They contain an oligosaccharide group, meaning a short chain of sugars. This is the head group, and at least one molecule of sialic acid. Because of this sialic acid group, gangliocytes actually carry a negative charge, and they're especially important in cell recognition and immune responses. And we can see all of this laid out in the figure below. So this helps illustrate the structural differences, starting with simple sphingolipids like ceramide and progressing into complex forms like cerebrocytes and gangliocytes, each with more elaborate head groups. We can also point out sphingomyelin, and it's highlighted with its phosphate-based head group, while something like gangliocytes, they clearly show the sialic acid unit in pink, indicating their negative charge. Finally, there's one last class of structural lipids we need to talk about, and that is waxes. Waxes are esters that are formed by combining a long-chain fatty acid with a long-chain alcohol. And because of these long hydrophobic chains, waxes are extremely nonpolar and they're solid at room temperature. Biologically, their main role is protection, and this applies to both plants and animals. In plants, waxes are secreted onto surfaces like leaves and stems to form a protective coating. And this coating helps reduce water loss by evaporation, and it also acts as a barrier against parasites and pathogens. In animals, waxes are used to prevent dehydration by repelling water. They also serve as lubricants, for example, in skin and fur, or in specialized secretions like earwax in humans. While waxes might not be as central to membrane structure as phospholipids or sphingolipids, they're still essential for protecting biological surfaces in a wide range of organisms. Now, before we move on to our second objective on signaling lipids, let's take a moment to pull everything together using this flowchart. This diagram summarizes only the classes of lipids you're responsible for on the MCAT. There are other lipids and there are other subtypes that you might see in advanced biochemistry, but for test day, this captures all of the major players. At the top, we start with the broad class of lipids, which we divide into 3 main functional categories. We have structural lipids, which make up cell membranes and protective barriers. We covered this in the first objective. Then there are signaling lipids, which act as chemical messengers. We're going to cover that in the second objective. And then there is energy storage lipids, which store fuel for the body, and that is what we're going to cover in the third objective. Now, under structural lipids, we've learned about phospholipids, glycolipids, and waxes. Phospholipids, they can have either a glycerol backbone or a sphingosine backbone. If the backbone is glycerol, we call them glycerophospholipids, and they contain 2 fatty acids and a phosphate plus an alcohol head group. If the backbone is sphingosine, we call them sphingophospholipids, and they also contain a phosphate head group and an alcohol, and of course, 2 fatty acid chains. Next, we have glycolipids, which are lipids with sugar head groups instead of phosphate. The main type of glycolipids we need to know for the MCAT are glycosphingolipids, and they have a sphingosine backbone, 2 fatty acid chains, and a sugar-based head group with no phosphate. These can be further classified based on the complexity of the sugar. Cerebrocytes have 1 single sugar, globocytes have 2 or more sugars, and gangliocytes have complex oligosaccharides and sialic acid, which gives them a negative charge. So again, gangliocytes have complex oligosaccharides and sialic acid. It's also worth noting here that glycosphingolipids are not the only type of glycolipids in biology. For example, there are glyceroglycolipids. They are found in plants and bacteria, but we don't need to elaborate on that right now. Finally, we also covered waxes, which are formed from a long-chain fatty acid and a long-chain alcohol. These are highly hydrophobic, and they're used by plants and animals for waterproofing and protection. So with that, we've completed the first objective, and we can move into the second objective, which will focus on signaling lipids. So far, we've been talking about lipids as structural components of membranes, but lipids also play active roles in cellular communication. They're involved in signaling pathways, hormone synthesis, and even act as coenzymes. One important family of signaling lipids is the terpenes. Terpenes are lipids that are made of repeating units of a 5-carbon molecule called isoprene. Isoprene has a molecular formula of C5H8. So these terpene molecules, they're built in multiples of 5 carbon atoms. So you'll find terpenes with carbon skeletons of 10, 15, 20, and so on. These are naturally occurring in many plants, and they have distinctive scents like citrus, pine, and herbs. You might recognize some of the examples that are shown here. Further, terpenes are important because they serve as precursors to a wide variety of biologically significant molecules, including steroids and fat-soluble vitamins like A, D, E, and K. Now, we can classify terpenes based on the number of isoprene units they contain. A monoterpene has 2 isoprene units. Sesquiterpenes, they have 3. Diterpenes have 4. Triterpenes have 6. Tetraterpenes have 8. And it continues up to polyterpenes, which are large chains. Now, terpenoids, they're slightly different. These are chemically modified terpenes. That means oxygen-containing functional groups may be added, or the carbon skeleton may be rearranged. But despite these changes, terpenoids still come from the terpene-based structure. Both terpenes and terpenoids fall under the category of signaling lipids because of their involvement in hormone synthesis and cell communication. And they're particularly important as intermediates or precursors in pathways that produce hormones like testosterone, cortisol, or estrogen, which leads us to discuss steroids. Steroids have a very distinct core structure. They're made up of 3 cyclohexane rings and 1 cyclopentane ring, and they're all fused together in a rigid 4-ring system that you can see right here. This fused ring system is what defines steroids. And while the core is always the same, the oxidation state and the functional groups attached to it, they can vary widely. This variability is what gives rise to the diversity of steroid hormones like testosterone, estrogen, and cortisol. These hormones differ in their side chains, but they all share the same base structure. Now, here's what makes steroid hormones so powerful. First, they bind to high affinity receptors, meaning their target receptors are very specific to them. Second, they work at low concentrations, so even small amounts can trigger major physiological effects. Finally, they affect gene expression and metabolism by directly entering cells and then interacting with intracellular receptors often found in the nucleus. Then, in addition to hormone signaling, steroids have structural roles too, especially cholesterol. Cholesterol is a steroid that is critical for membrane fluidity and stability. It inserts between phospholipids in the membrane where it can help regulate how rigid or flexible the membrane is. Cholesterol also serves as a precursor for many other important molecules, things like steroid hormones, vitamin D, and bile acids. Next up is prostaglandins. Prostaglandins are autocrine and paracrine signaling molecules, meaning they act locally, either on the same cell that produces them or on nearby cells. Prostaglandins, they're derived from arachidonic acid, and they work by regulating cyclic AMP levels inside the cell. They play key roles in smooth muscle contraction, in body temperature regulation, in the sleep-wake cycle, and also inflammation, fever, and pain. They are actually one of the main targets of drugs like non-steroidal anti-inflammatory drugs, which work by inhibiting prostaglandin synthesis to reduce your pain and swelling. Finally, we are going to discuss fat-soluble vitamins. A vitamin is defined as an essential nutrient that the body cannot synthesize in sufficient quantities. That means it has to be obtained through your diet. Now, there are 4 fat-soluble vitamins that you need to know for the MCAT: A, D, E, and K. And here's a quick rundown of each. Vitamin A includes compounds like retinol, retinal, and retinoic acid. These are essential for vision, cell growth, bone development, and immune health. And good sources include beef, liver, and dairy products. Provitamin A refers to carotenoids, which are antioxidants that are found in colorful vegetables like carrots and spinach. These can be converted into active vitamin A forms in the body. Vitamin D includes D2 and D3 forms. This is important for bone metabolism as well as immune health. And you can get it from cod, liver oil, fatty fish, and dairy, or you can synthesize it in the skin with sunlight. Vitamin E acts as a powerful antioxidant as well, and it prevents and protects membranes from oxidative damage. It's found in olive oil, nuts, and seeds. Finally, we have vitamin K, including phylloquinone and menaquinones, and this is necessary for activating blood clotting factors and bone proteins. It's found in leafy greens, legumes, and other plant-based foods. With that, we've completed objective 2, and now we can move into our third and final objective titled energy storage. The main lipids that are responsible for storing energy in the body are called triacylglycerols, also known as triglycerides. These are specifically built for long-term energy storage, and here's why they're so efficient. First, the carbon atoms in fatty acids, they are more reduced than the carbons in sugars. So that means they yield more energy per unit when they're oxidized. Second, triacylglycerols are hydrophobic, so they don't carry water weight with them. This makes them much more compact and efficient for storage compared to carbohydrates like glycogen, which are heavily hydrated. Now, structurally, a triacylglycerol molecule consists of 3 fatty acid chains that are each esterified to a glycerol backbone. This is a condensation reaction. So for each ester bond that forms, a molecule of water is released. And you can see that process in the diagram. Glycerol plus 3 fatty acids yields a triglyceride and 3 water molecules, with the linkages shown as ester bonds. In the body, these triacylglycerols are stored in specialized fat cells. These cells are packed with triglyceride droplets, and they serve as energy reserves that can be broken down when they're needed. Let's now take a closer look at free fatty acids and how they connect to what we just learned about triacylglycerols. A free fatty acid is simply a fatty acid that is not attached to anything. It's not part of a triglyceride or a phospholipid. It exists in the body in its unesterified form, meaning the carboxylic acid group is still intact. And that free carboxylate group makes the molecule amphipathic. That allows it to interact with both water and lipid environments. Now, here's the connection. When we break down triacylglycerols, we release these free fatty acids. And one of the most well-known methods for doing this is called saponification. Saponification is the base catalyzed hydrolysis of ester bonds. So in other words, we use a strong base like sodium hydroxide or potassium hydroxide to break the ester linkages in a triglyceride. And this reaction releases glycerol and 3 fatty acid salts, which we call soap. So in this reaction diagram, you see that the triglyceride reacts with 3 equivalents of sodium hydroxide, and the result is 1 glycerol molecule and 3 soap molecules, which are sodium salts of fatty acids. Now here's where structure meets function. When you place these soap molecules in aqueous solution, they're not just going to float around randomly. Because they're amphipathic with a polar head and a nonpolar tail, they're going to organize themselves into spherical structures called micelles. In a micelle, the hydrophilic or polar heads, they face outward and they interact with the surrounding water, whereas the nonpolar or the hydrophobic tails, they face inward. They're shielded from the water and they're able to trap oils or other nonpolar substances inside. This is exactly how soap works. It forms micelles that surround greasy nonpolar particles, and they allow them to be rinsed away in water. So to tie it all together, triacylglycerols store energy efficiently in the form of fatty acid chains. When broken down through saponification, they release free fatty acids and glycerol. The free fatty acids, when converted into salts, become soaps, which can self-assemble into micelles that function as emulsifiers in water. And with that, we've completed this chapter. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.